Characterization of bacterial culture in agar plate. Bacterial colonial morphology, or cultural characteristics, is a key factor in the identification of bacteria. Bacterial growth characteristics such as size, shape, margin, form, texture, etc. Pigmentation of colony. Surface characteristics. Shape of colony. Size of colonies.
We have many colony morphology or characteristics of bacterial cultures. So we have size of the colonies, preferably in millimeter. We have the punctiform or pinpoint, which is less than 1 millimeter. The small size of colony is 1 to 2 millimeters. The medium is 3 to 4 millimeters, and large is 5 to 5 millimeters and above, or more than. This example of pinpoint size is the Streptococcus pyogenes, which has a pinpoint size of 0.5 to 1 millimeter. And we have in the lower image is the many bacterial culture that is a size large size of colony, which measures 5 millimeter. Then we have the margin of colonies. We have the entire. It is smooth and does not have notches. And our example for this is Streptococcus pneumoniae. As you can see, the margin, the entire part of the bacterial culture is smooth and it is round. It does not have any curves or notches or deep indentations. Then we have the angulate. For example, in angulate is Mycobacterium tuberculosis. It has a wavy and has a shallow indentations. This example of an angulate. Then we have the lobate. The lobate has a wavy and much more deeper indentations than the angulate. Then we have the rose or serrate. It is irregularly notched. As you can see, it has an irregular notch in its margin. Then we have the filamentous. It has a long, irregularly placed or interwoven threads or filaments in the margin of the bacteria. Then we have the curl. It has a parallel chains and wavy strands. As you can see, the parallel part of the margins, and it's not smooth. Yeah. Then we have the elevation of colonies. We have the flat elevation. For example, the Pseudomonas aeruginosa. Then we have the raised. Elevation of bacterial culture. Then we have the ambonic, raised with a thicker center than margins. It's also known as the knob-like elevation. Then we have the convex. It's like a segment of a circle but flattened. It's like a semicircle. It's convex. Then we have the pulvinate. It's also known as a cushion-shaped elevation of the bacteria. And then we have the odor. Each bacterial culture has different odors. Some has no, but others resembles the smell of fruity grape-like. For example, in Pseudomonas aeruginosa. And in Proteus species naman, it resembles the smell of burnt chocolate. The pigment of surrounding medium, we have the Salmonella type A as an example. In blood agar, it is yellowish. Its pigment color is yellowish. Then in Machanchoid agar, it is colorless. In deoxycholate citrate agar, colonies show black heads. And in Wilson and Blair bismuth sulfate agar, it's jet black colonies with metallic sheen.
Characterization of bacterial culture in agar's lens. Agar's lens are used for storing pure cultures for a moderately long term. There is a minimal risk of contamination or of losing the culture due to the medium drying out because the small volume of air inside the tube and narrow opening combine to limit water loss and pressure to outside air, including dust and other particles. Amount of growth. Pseudomonas aeruginosa is a common free-living gram-negative bacterium that can cause significant disease as an opportunistic pathogen. Rapid growth, facile genetics, and a large suite of virulence-related phenotypes make P. aeruginosa a common model organism to study gram-negative opportunistic pathogens and basic microbiology. Color of colony. In a slant agar culture, the color of the colony is red because of the presence of lactose, glucose, and sucrose. The P. aeruginosa is a lactose fermenter responsible for the color red. In a Machanchi agar medium or slant, the colonies of Pseudomonas aeruginosa are colorless due to the lack of lactose fermentation, which is of great importance in differentiating P. aeruginosa from other bacteria present in the specimen. Pigment production in the culture of Pseudomonas aeruginosa. In the culture medium, P. aeruginosa produces mainly 4 types of pigments, which diffuse off into the surrounding medium and imparts a characteristic color to the medium indicating the suspected growth of Pseudomonas aeruginosa. These are described as pyocyanic bluish green, fluorescent, greenish yellow, pyroubicin, reddish brown, pyroluminine, brown to black. Of the 4 types of pigments, only the pyocyanin is important from the diagnostic point of view, as pyocyanic is the only pigment that is not produced by any other species of Pseudomonas but only by the Pseudomonas aeruginosa is imparting characteristic color to the medium representing consistency. Viewed by transmitted light, the colonies were smooth, convex, opaque, glistening, entire, and psychocyogenic. They were butyrous to the touch and produced a uniform turbidity when suspended in a 0.85 of sodium chloride. Optical characteristics. The colonies of the original culture of P. aeruginosa had a raised, opaque, glistening center bordered by a depressed, less opaque, dull concentric ring. The colony mass tempered gradually from the outer edge of this ring to irregular translucent form. It is found out that it is in a filiform. It is a uniform along the line of inoculation. Odor, absent or resembling. In an ordinary medium like nutrient agar medium, Pseudomonas aeruginosa produces a characteristic sweetish odor or a distinct musty or earthy smell in the medium. Pseudomonas aeruginosa grown in media is also being characterized by grape-like smell. Being a non-fermenter, the bacterium is also associated with acid produced rather than gases, which are commonly associated with fermenting bacteria. Whereas in its medium change, none or color change, it is none.
Characterization of bacterial culture in nutrient broth. Broth cultures are liquid culture used to grow bacteria in laboratories. To create a broth culture, a scientist begins with a sterile liquid growth medium. The medium is inoculated with bacteria and placed in an incubator at the appropriate temperature. After a certain amount of time has passed, the broth becomes cloudy from the increased number of microbes. Examination of broth culture must include first is the surface growth. As you can see in the picture, there is no visible growth in here. Next is the ring and pellicle. This is an example of 48-hour-old Bacillus thuringiensis floating biofilm. The biofilm was formed in a glass tube at 30 degrees Celsius in HCT medium. The culture medium was slowly discarded by pipetting, resulting in a separation of the ring and the pellicle. This is the ring, and it is a thick structure which sticks to the culture container at the upper margin adherent to glass. Next is the pellicle. It is under the ring, and it extends over the whole liquid surface and joins the ring. It grows in the form of a continuous or uninterrupted sheet over the surface. This is Streptococcus, and it shows the growth of flocculent. And as you can see, there are small masses of growth floating in the culture fluid. Next is the membranous. It is a thin coherent growth like a membrane. Next, turbidity. It shows the surface growth of bacteria is cloudy. Next is the granular. It can be seen in the surface growth as small particles. And last is the flaky, and it is seen as a large particles. Next is the amount of growth. It can be scanty, moderate, abundant, or no visible growth at all. Sediment. It is a mass of bacteria at the bottom of the suspension. It can be classified as granular, which has small granules, flocculent, small masses of growth of various shapes, flaky, separate flakes, viscid, sediment rises as a coherent swirl. Lastly, their pigmentation. These are isolated bacterium. The B1 was yellow pigmented while the B2 was orange pigmented. Both were gram-positive cocci with smooth colony.
Bacterial culture in agar slants can be characterized into three major categories. Number 1 is the amount of growth, second is the form of growth, and the third and last is liquefaction. Amount of growth. It can be none, scanty, moderate, and abundant. Form of growth. In the form of growth, it can be filiform, beaded, papillate, villous, or arborescent. Liquefaction. In liquefaction, it can be non-crateriform, naviform, invindibule, saccate, and stratiform.